Welcome to our discussion on hypersensitivity reactions. These are immune responses that go beyond what's necessary, causing harm instead of protection. There are four main types, each with distinct mechanisms. Type 1 involves allergens triggering IgE antibodies on mast cells, leading to immediate reactions. Type 2 is about antibodies targeting cells directly, causing damage. Type 3 centers on immune complexes that activate neutrophils and cause tissue injury. Type 4 is a delayed response where T cells release cytokines to affect target cells. Understanding these types helps us grasp how immune responses can sometimes backfire. Let's talk about allergy, which is a classic example of hypersensitivity. Basically, it's when our immune system reacts too strongly to something harmless, like pollen or certain foods. This reaction happens in two stages. First, the initial exposure sensitizes the immune system, and then the second exposure triggers a more intense, sometimes harmful response. So allergy is really an exaggerated, unwanted immune reaction that can cause symptoms ranging from mild irritation to severe problems. Now, the Gell and Coombs classification organizes hypersensitivity into four types based on how the immune system reacts. Type 1 is immediate and IgE-mediated, causing things like hay fever or anaphylaxis within minutes. Type 2 involves antibodies attacking cells directly, causing damage. Type 3 centers on immune complexes that activate neutrophils and cause tissue injury. Type 4 is a delayed response where T cells release cytokines to affect target cells. Understanding these types helps us grasp how immune responses can sometimes backfire. Let's focus on type 1 hypersensitivity. This is the classic allergic reaction that happens quickly after exposure. It's driven by IgE antibodies, and some people have a genetic tendency called atopy to produce more IgE. The allergen can enter the body in many ways, breathing it in, eating it, or through the skin. The reaction can be localized, like in hay fever, or systemic, which can be life-threatening. Symptoms vary widely, so recognizing this type is crucial for timely treatment. Let's dive into how type 1 hypersensitivity works. When someone is first exposed to an allergen, their immune system processes it and produces IgE antibodies specific to that allergen. These antibodies latch onto mast cells and basophils, priming them. Upon re-exposure, the allergen binds to these IgE antibodies, causing the mast cells to release chemicals like histamine. This release leads to the familiar allergy symptoms, itching, swelling, and in severe cases, anaphylaxis. Understanding this sequence is key to managing allergic responses effectively. Digging deeper into type 1 hypersensitivity, reactions happen when someone who's already sensitized encounters the allergen again. Mast cells release substances like histamine, which dilates blood vessels, and leukotrienes, which tighten bronchial muscles. This explains why allergies can cause swelling and breathing difficulties. Importantly, penicillin allergies are a common example to watch for. Treatments focus on counteracting these effects, using epinephrine to reverse severe reactions, antihistamines to block histamine, and corticosteroids to reduce inflammation. Type 2 hypersensitivity is all about antibodies directly causing cell damage. This includes situations like blood transfusion reactions, where antibodies attack mismatched red blood cells, leading to their destruction. Another example is hemolytic disease of the newborn, where maternal antibodies target fetal red blood cells. There's also antibody-dependent cell-mediated cytotoxicity, where antibodies attract immune cells to kill targeted cells. These mechanisms show how antibodies, while protective, can sometimes harm the body's own cells. Type 2 hypersensitivity is all about antibodies directly causing cell damage. This includes situations like blood transfusion reactions, where antibodies attack mismatched red blood cells, leading to their destruction. Another example is hemolytic disease of the newborn, where maternal antibodies target fetal red blood cells. There's also antibody-dependent cell-mediated cytotoxicity, where antibodies attract immune cells to kill targeted cells. These mechanisms show how antibodies, while protective, can sometimes harm the body's own cells. Type 3 hypersensitivity involves immune complexes, clusters of antigens bound to antibodies that deposit in tissues like blood vessel walls and kidneys. These deposits activate complement, attracting neutrophils that release enzymes and reactive molecules, causing tissue damage. This process underlies conditions like serum sickness and the Arthus reaction. So the key idea is that these immune complexes trigger inflammation and injury where they accumulate, highlighting the importance of immune regulation. Looking at the mechanism behind type 3 hypersensitivity, small immune complexes form when antibodies bind antigens. These complexes can grow and deposit in tissues, activating complement proteins like C1 and C3b. This activation produces C5a, which calls neutrophils to the site. The neutrophils stick to the tissue and release harmful enzymes and reactive oxygen species, damaging the local cells. This cascade explains how immune complexes lead to inflammation and tissue injury, especially in blood vessels and kidneys. Type 4 hypersensitivity is unique because it doesn't involve antibodies. Instead, it's a delayed reaction driven by T cells. Macrophages present antigens to helper T cells, which then activate cytotoxic T cells to attack target cells. This process takes days to develop and is responsible for conditions like contact dermatitis, tuberculin skin tests, and graft rejection. Understanding this cell-mediated response is important since it operates differently from antibody-driven hypersensitivities. In the sensitization phase of type 4 hypersensitivity, antigen-presenting cells like macrophages or Langerhans cells process intracellular bacteria and present their antigens to helper T cells. This interaction activates Th1 cells and sometimes CD8 cells, which become the mediators of delayed type hypersensitivity. This step sets the stage for the immune system to respond more aggressively upon subsequent exposures, emphasizing the cellular basis of this reaction type. During the effector phase of type 4 hypersensitivity, sensitized Th1 cells interact with resting macrophages, activating them through cytokine release. These cytokines include interferon gamma, tumor necrosis factor beta, and several interleukins, which enhance macrophage functions like antigen presentation and production of reactive oxygen species. Activated macrophages then contribute to tissue damage and inflammation. This cellular dialogue explains the delayed and sustained nature of type 4 hypersensitivity reactions. Let's review the 4 hypersensitivity types together. Type 1 is immediate and IgE-mediated, causing rapid allergic reactions like asthma or anaphylaxis. Type 2 involves antibodies destroying or altering cells, leading to conditions like hemolytic anemia. Type 3 is driven by immune complexes that deposit in tissues, triggering inflammation seen in diseases like lupus. Type 4 is cell-mediated and delayed, with T cells activating macrophages or killing cells directly, causing reactions like contact dermatitis. Recognizing these patterns helps us understand and manage immune-related diseases. Type 5 hypersensitivity is a bit different from type 2 because here, antibodies stimulate rather than destroy target cells. A key example is Graves' disease, where antibodies mimic thyroid-stimulating hormone and cause excessive thyroid hormone production, leading to hyperthyroidism. This stimulation occurs due to antibodies binding TSH receptors, sometimes triggered by infections like Yersinia enterocoliticus. Myasthenia gravis is another example where antibodies interfere with muscle function. This highlights how antibodies can alter cell behavior beyond just causing damage.